My name is Sandra Schachat. I am a research student in the departments of paleobiology and entomology, and I have been here since 2009. Today I'm going to talk about the evolution of wing pattern in insects in the order Lepidoptera, so that would be butterflies and moths. And wing patterns in Lepidoptera have interested evolutionary biologists going all the way back to Charles Darwin, and I'm sure that many of us, even during our childhoods, have noticed the beautiful wing patterns of insects in this order. And given that these wing patterns have interested so many people for such a long time, perhaps it's a bit surprising that there are still some major gaps in our knowledge of these wing patterns. And I'm going to address the two main gaps today. The first is that we still don't understand the evolutionary history of wing patterns among the Micropterix. These are your typical tiny brown moths. So here on the screen, you can see illustrations of wing patterns in two species belonging to very distantly related lineages. These wing patterns look very similar, and the question is whether this might represent an ancestral state that originated once, or whether these sorts of wing patterns might have originated multiple times independently, perhaps according to different developmental pathways. We still don't know. And then the second main gap in our current knowledge is that we don't understand how the more complex wing patterns that we see in butterflies and their close relatives originated from the simpler wing patterns that we typically see in tiny brown moths. So I'm going to address this first gap right now, the evolutionary history in tiny brown moths. And here on the screen you can see an illustration of a model that was proposed that does have the potential to be used to designate homologies and reconstruct evolutionary history for tiny brown moths. I call this the wing margin model. And if you focus on the wing margin up top that's highlighted in red, you'll see that where each ancestral vein terminates along that margin, those veins are each surrounded by one band of color. There's alternating dark and light brown bands. Now, this model was developed based on observations of one single taxonomic family of moths, the Tortricidae, but there's well over 100 families. And so for my master's thesis, my first goal was to evaluate whether this model also holds for other distantly related families of moths. I focused on a family called Micropterix. These are the earliest diverging moths that are alive today. Here's one example, the species Micropterix schaefferi, and as you can see, the wing margin model does hold for this species because where all of the ancestral veins terminate along that upper costal margin of the wing, we have one band of color surrounding each vein with these alternating dark and light bands. Now here you can see another species from that same family Micropterix. This is Sabatinca doroxena, and at first glance it appears that we have pretty much the same exact thing going on in this species. We have these alternating dark and light bands of color surrounding each ancestral vein at the wing margin. But if you focus on the vein that I've highlighted with an arrow here, the first branch of the subcostal vein, in Sabatinca, this vein is surrounded by a light brown band of color, whereas in Micropterix, this vein is surrounded by a dark brown band of color. These are very closely related species in the same family, and yet what's dark in Micropterix is light in Sabatinca and vice versa. And so this suggests that color can be misleading and should not be used to designate homologies, and is really not indicative of evolutionary history. What's most important, if we want to reconstruct how this variety of wing pattern evolved, is the location of these wing pattern elements, particularly with relation to veins. So we do have these two alternating series of bands, but either series could become light or dark. We should focus on their location, not their color. And now moving on to the origin of the more complex wing patterns that we see in butterfly and their kin. These wing patterns are comprised of symmetry systems. So as opposed to one single band of just one color, we have concentric color, multiple bands in the same wing pattern element. So typical butterfly wing patterns have 3 symmetry systems. Each of these is indicated by an arrow on the slide. Now, there are multiple hypotheses for how these symmetry systems could have originated, but at present, those hypotheses are purely speculative. None of them are supported by any empirical data. So to get at this question, I wanted to go back to the tiny brown moths that I began with for my master's thesis. So I'm going to show you the wing pattern of Sabatinca doroxena, a tiny brown moth that I showed a few slides back, and I'm going to plot that wing pattern onto the wing of a hypothetical butterfly. Now, there's any number of ways that you could manipulate these patterns when you're transferring them from one Lepidopteran to another, because these wings vary tremendously in size and shape. But there's really only one way to transfer, quote-unquote, these wing patterns if we wish to preserve the relationship between wing pattern and wing venation. And I do want to preserve that relationship because my earlier results had suggested that it is very important. And just to orient you, the two red arrows that you see on this slide point to the same wing vein, which shows just how drastic these differences in wing shape are. So this is my result if I plot the wing pattern of Sabatinca doroxena onto the wing of a hypothetical nymphalid butterfly. And this is the nymphalid ground plan, which shows the essential elements of wing patterns in butterflies in the family Nymphalidae. And as you can see, these are very similar, which suggests that this relationship between wing pattern and wing venation can be very helpful in allowing us to reconstruct the evolution of all of these wing patterns, because it controls for changes in wing size and wing shape and allows us to compare the patterns of tiny brown moths and butterflies. Thank you very much. Within a species, the patterns can vary, particularly in the genus Micropterix, which I had showed earlier. So when I studied these moths, I only showed one example per slide, but I would look at many, many individuals in both wings. The patterns can vary between the two forewings on one single individual, but I never found any variations of the wing margin, or I never found any violations of the wing margin model, simply variations of it. I became interested in insects very slowly, just reading about popular science, and whenever you'd read about some very strange evolutionary innovation, it was often in an insect. And I also had studied art history, and so I essentially think of my master's thesis as the art history of insect wings.
Is there a mechanical reason for that, or a physiological reason, or is it just something you observe?
It's something we observe. And so what's tricky about the relationship between veins and color pattern in moths is that veins can still influence the development of these color patterns, even if an ancestral or plesiomorphic vein is not expressed in the adult wing. And we know that we know that that is the case based on observations of thousands of species of butterflies. How this actually happens is still unknown. And unfortunately, with the Micropterigidae, there's no lab colonies at all. People tried for years and weren't able to establish any, so that precludes the sort of molecular work that might shed light on that. I probably create a lot more illustrations than I need to, and that's because I think it's fun. Other than that, I think that perhaps the way that I arrive at my research questions might be driven by my interest in visual evidence, but I would say that my methods are pretty standard for insect morphology, just with more pictures.
These interactions, male sexual dimorphism, etc.
So the question is, which proportion, more or less, of these wing patterns are? I would say that it's probably mostly crypsis. Certainly, nocturnality is very, very common in Lepidoptera. With that being said, the Micropterigidae that I showed are day flying. So I would say that you usually have crypsis. Insects don't see as well as far as spatial resolution as we do, for example. They see very poorly. So I think that you have a lot of crypsis, some wing patterns that mostly function to startle predators, and I would think that it's relatively uncommon for intraspecific communication to occur via wing patterns. Pheromones are very common for intraspecific communication.